Hello, and welcome to episode number 2 of Antibodies, the official podcast of the Antibody Society. I'm your host, Jonathan Davis, founder of the consultancy Creative Antibodies. On this show, we plan to explore the science, stories, and people driving antibody discovery, engineering, and therapeutics with a mixture of personal stories and technical insights that we hope will give everyone a reason to listen. For this episode, which is being recorded live at the Antibody Engineering and Therapeutics Conference in San Diego, I am delighted to be joined by a leader in the field of antibody engineering, John Desjarlais, who currently holds the roles of Executive Vice President for Research and Chief Scientific Officer at Xencor. Since joining Xencor in 2001, Dr. Desjarlais has overseen the company's engineering, discovery, and preclinical work on antibodies and other proteins. With his team, John has developed several novel technologies for the optimization of antibodies and other proteins, including a platform for generation of bispecific antibodies, and has led the discovery of multiple therapeutic antibody candidates, including CD3 bispecific antibodies, checkpoint bispecifics, and cytokine Fc fusions for treatment of a variety of diseases. Prior to Xencor, John was an assistant professor of chemistry at Penn State University for 4 years, where he developed and tested computational methods for the de novo design of proteins. He began his work in the field of protein design as Jane Coffin Child's Fellow at UC Berkeley. John holds a PhD in biophysics from the Johns Hopkins University and a BS degree in physics from the University of Massachusetts Amherst. Welcome to the podcast, John.
Well, thanks, Jonathan. This is fun. This is good to be here.
Great. But before we get into all the science, I want to go back before your science career took off. Here on Antibodies, we talked about music and the connection between music and science, and like me, you were a serious musician back in the day. Tell us briefly about that.
I'd like to say in a former life, I was a classical trombonist. That was my aspiration throughout high school. And in fact, my first year of college, I was a music major, training to be a classical trombonist. Also played a lot of jazz trombone. And as you know, I've dabbled with various musical instruments throughout the rest of my life, teaching myself trumpet, congas, guitar, and even some harmonica.
Right, which is an instrument that I play as well. So I think the music-science connection is really incredible. It seems to be such a common theme. Trombone it's a good instrument. It's a really good instrument.
It's a difficult instrument.
It is. I took it up once very briefly, and I did not keep with it. So, okay, now to the antibody portion of the program. Let's start with your history in antibody engineering, which has been long and fruitful. Can you tell us how you got started? What has made antibodies hold your interest for over 2 decades?
Yeah, sure. In fact, it was never my intention to work on antibodies. When I was at Penn State, I was working on the kind of stuff that David Baker was doing. I was working on, computational protein design. That was also the genesis of the Xencor company concept. So when I joined Xencor in 2001, my first project was working on optimizing a plant enzyme to do something better in plants. And so we were focused on, engineering industrial enzymes and other kinds of enzymes. The antibody aspect happened when somebody came to interview at Xencor for a position, and he had been a postdoc working down the hallway from Jeff Ravitch, who was very interested in how antibodies engage Fc gamma receptors and was just beginning to explore mutating the Fc domain to enhance activity or engagement of the various Fc gamma receptors. And we thought, that's a problem we can tackle. Sometime around then, I also hired Greg Lazar, who's another leader in the field, and he made some mutations that are now being used more and more frequently. There are several approved antibodies and probably more to come.
Yes, one thing is as they go off patent, 20 years after they get discovered, people start using them a lot.
That's right. Yeah, people are using them far more frequently now versus back when we actually invented them.
But that's one of the wonderful things about this. I think that it gives you a chance. You can make an invention, and your company and maybe you can make good money off it while it's new and proprietary. And then if it's something that has staying power, it gets used by so many people, and you can point to hundreds of drugs someday maybe that said that drug is using my invention. And
that's absolutely, yeah, there's a bittersweet aspect to it, right? So the bitter part is you didn't make the money that you could have for your company early days through licensing, but the sweet part is actually, like you said, the longevity of these things. So not only are people using those high ADCC mutations in various molecules, but our LS substitutions that we call extend are being used to enhance antibody half-life for, in fact, it was in sotrovimab, one of the leading anti-COVID antibodies from Vir, and is in a number of other approved antibodies, and people are applying that more and more these days.
Yeah, I think it's in a lot of antibodies that are in discovery and development at this point. So, well, we all get into biotech, though, to have an impact on science in the pharma industry and ultimately on patients. So you've had a really impactful career. I mean, Xencor really made a lot of the early advances in engineering the Fc to make an antibody a better drug, because nature evolved the antibody to be part of your immune system. And when we have a drug, we want it to do something very specific. Your immune system is more general, right? So you figured out some ways of making the antibodies better drugs. And like we just were saying, these are important things that have been used now by a number of people. What would you say is the highlights of your career of antibody engineering?
I would say the first highlight, the real big milestone is you discover these antibodies, these antibody platforms, and particularly when you're a starting biotech company just getting into clinical development, it could be years before you see any impact of all that hard work on treating a cancer patient or something like that. And so when we first started seeing responses with some of the molecules we made, either in lymphoma patients or Hodgkin lymphoma patients, that's a big highlight. I would say probably, the most exciting thing for me ever, we developed a number of FC mutation technologies. I mentioned the high ADCC, I mentioned the LS. Another one was the mutations that enhance affinity for Fc gamma R2b, the inhibitory receptor. That led to this molecule that is now called obexelimab, which targets CD19 and engages Fc gamma R2b on B cells. And it actually shuts them off. It turns them off, doesn't deplete them, but stops them from being activated. We were super excited about that molecule. I'm very proud of that molecule. And at the time, it was partnered with Amgen, and they went into a healthy volunteer study. And just as a test of whether or not the molecule works, they immunized them with KLH. And when I saw that data set showing, about a log reduction of the ability of humans to produce antibodies against KLH, that was a real special moment for me. In fact, I still have the prism plot of that data tacked up on my my tack board in my office.
Oh, that's fantastic. And I just want to help for listeners who don't know what KLH stands for. So it's keyhole limpet
Hemocyanin.
Hemocyanin, thank you. Okay, even I don't know. It's this big protein out of a very obscure little sea creature that, for whatever reason, is very, very immunogenic in all animals. So it's become the standard. And for one thing, no one's been exposed to it. So that there's not going to be pre-existing antibodies to this thing.
That's right.
Right.
Yeah.
So it's a useful tool, and that's what KLH is.
Yeah, and in demonstrating that we could blunt that response, of course, means that the molecule potentially has applicability for autoimmune diseases where antibodies are a major driver. And I should mention that that molecule obexelimab ultimately ended up in the hands of a company called Zenus Bio. And in fact, they are about to finish up a phase 3 study using obexelimab for the treatment of IG4-related disease.
Wow, that's good. Phase 3. So do you think it will end up getting approved?
I think it will, yeah. In fact, they just released some data that obexelimab also works in multiple sclerosis. I think they've guided that they're not going to be pursuing that because it's a tough indication to develop in, but it was fantastic proof of concept.
Will that approach be useful for a wide range of autoimmunity, you think? This idea of shutting down B cells?
Yeah, I mean, the people have explored that idea. I mean, one idea we had gotten excited about, and I think some companies have actually pursued, is the idea of stringing a autoantigen onto that Fc domain so that you would specifically inhibit B cells who have the B cell receptor that recognizes that autoantigen.
I actually remember a talk you gave maybe 15 years ago when you first described that. And I remember thinking, wow, that is really smart. That's a great idea.
Yeah, we actually also applied it to anti-IgE.
Mm-hmm.
And that was a program that was, as we think about some of the challenges in biotech and some of the peaks and disappointments, that's a great example. Everything about that molecule was fantastic preclinically. From the first in vitro data set and the first mouse studies, just outrageously exciting ability to inhibit the production of IgE as well as sequester IgE. The non-human primate studies, all looked great. We got into humans, and for reasons we still don't understand, that molecule leads to a transient reduction in platelets. And it's reversible. The platelets go down and they come back up. But at the time, it was kind of a safety concern for us. Big disappointment there with an otherwise fascinating molecule.
Well, that's part of the world, I think, of biotech and pharma is things don't always work the way you expect when you put them into people. That's why we have to have trials.
Yeah, we're still hopeful that that molecule has some legs. We're still looking at it. There's some folks that are still interested in that mechanism. And I think based on more recent clinical data in the hands of a partner, it looks like the platelet reduction is not actually a problem.
And would it be competitive with Xolair?
Yeah, the whole idea with the molecules, not only does it prevent the production of IgE because it can stop those B cells from turning into plasma cells. The other unexpected mechanism at the time, although if we had read the literature better and understood the biology of Fc gamma R2b better, it wouldn't have been surprising. Turns out something like 2/3 of the Fc gamma R2b in the body is in the liver as a scavenger receptor. So what happens when you dose either non-human primates or humans with this molecule, unlike Xolair, which blocks IgE and actually increases the total concentration of IgE, this molecule actually scrubs total IgE out of circulation. And so you don't have sort of the dosing challenges with this molecule that you would have with something like obexelimab.
So the antibody binds IgE and then gets picked up in the liver via the Fc gamma R2b enhancements.
Yeah, the L6, the liver sinusoidal endothelial cells.
That's interesting. So how did you come to embark on all these projects with the therapeutics and also the engineering? Did somebody tell you, we need to solve this problem, or did you just come up with ideas? Or
Yes.
was it that you were doing something else and
Right.
it was the old Asimov thing of, that's funny.
Right. No, I was thinking about that. So in the early days, it started out, like I mentioned earlier, that the genesis of our work on antibodies was, hey, let's see if we can tackle the Fc domain, right? And we made our ADCC mutations, and then we started thinking about, okay, what could we target? All right, CD30 was our first target. We also had explored CD20, the things where you can have impact in a particular disease. Then Fc engineering sort of became our calling card for that period of time from 2001 to 2010. So as we developed new Fc platforms like the enhanced Fc gamma R2b mutations or the long half-life mutations, now you've got, I guess, the proverbial hammer, and you've got to find out what to apply it to. And so the concept of obexelimab, that was just me sitting back trying to understand, how is it useful to inhibit a B cell? What do we target to inhibit a B cell? And so that led us to CD19. We looked at other targets, but CD19 was the one that worked. And then once we knew that worked, that also translated into targeting IgE because it's the same concept. And why not? Why not try it in another case? In many other cases, the ideas have just come out of brainstorming with Basil Dahiat, our CEO, where we can go with some of these things, or with my team. Ideas can come from any place. We actually invested heavily in, once we had a bispecific antibody platform, which we'll probably talk about in a few minutes, we invested heavily not only in T-cell engagers, but also checkpoint bispecifics. That was sort of opportunistic, right? Because, we knew that anti-PD-1s were having high impact. All these other checkpoints had been discovered, CTLA-4, LAG-3. And so then that was a game of, okay, we've got this great bispecific antibody platform. Let's start creating a bunch of these molecules, mixing and matching dual checkpoint inhibitors and seeing where that goes to. We ultimately got into clinical studies, had some really interesting results. I mean, we had efficacy, but that is a tough game, breaking into a market that's where, things like pembrolizumab and nivolumab are heavily entrenched, and showing that you can move the needle enough to have commercial viability.
Right. And you're not the only ones trying to do it. The number of PD-1 antibodies that companies are trying to develop is huge. So
that's right. Yeah. We're still hearing about some at this conference.
That's right. So you did mention bispecifics, and that may be what we should move on to next, because it's definitely a shared interest between you and me and a lot of people who are listeners, no doubt. So the bispecific field is just growing like crazy these days. There's so many more bispecific projects than there were just even a few years ago. So, a typical antibody, just to help the listeners, a typical antibody is Y-shaped molecule. The two ends of the Y are identical, and they bind the same thing. Multispecifics or bispecifics are antibodies with at least two different binding arms so they can bind multiple targets. And so that makes them drugs, not natural molecules. They're not the way antibodies normally are made. But the capability can be used to generate a biological effect through either proximity of the two things that it binds to, or increase the specificity of the antibody to cells that express both targets, or inhibit multiple redundant pathways, activate targets via clustering or pairing, or other things. So they've become a major part of the antibody landscape, and trispecifics are starting to emerge now in the discovery space. So, question for you. What do you see as the future for these technologies? Are there new uses that are only now being discovered?
Yeah, I think there are. In fact, I think we've heard about some of those in the conference. So Xencor, building off of concepts that were built at, other companies like Roche, we've used our bispecific antibodies to make T-cell engagers against not only liquid tumors like lymphoma and myeloma, but solid tumors as well. I think some of the new applications of bispecific antibodies are super fascinating, very early days. But for example, using the surface degraders, right? So the surface E3 ligases, we heard Jim Wells talk about that the other day, and I think he's got a company now that's pursuing that. But the ability to have, one side of your bispecific antibody engage a surface E3 ligase, whose job it is to degrade things, and have the other arm target something like EGFR, which is a cancer driver, and completely eliminate the EGFR from the surface of that cancer cell, that's a really exciting prospect. And I think there's a lot of things they could do there as well in terms of based on differential expression patterns of these surface ligases and promoting, selectivity for tumors versus normal tissue and so forth. I also think you mentioned trispecifics. I think one of the big challenges we have, even though T-cell engagers are really starting to make a dent in solid tumors, that's been successful for a very limited number of targets that are highly selective. Some of these are so-called cancer testes antigens where they're really only expressed in a few reproductive organs in the adult, but are highly overexpressed in tumors, or other targets like our EMPP3 for our kidney cancer program. But that's a short list of very specific targets. And, some people exploring masking technologies to increase specificity. But I also think that's where trispecific antibodies are going to fit in nicely. So if you have two targets that are expressed very strongly on a cancer cell, and you look for target pairs like that where they don't have overlapping expression on normal tissues, you can develop what's called a AND gate, where, in other words, my T cells will only kill if the tumor cell has target A and target B. So I think we're going to see a lot more of that emerging over the years. There are technical challenges to making trispecific antibodies in a way that they're highly developable and have good half-life and so forth. But I think there are also a lot of discovery technologies that can be applied to that problem.
So, yeah, you covered a lot there. Well, you mentioned solid tumors in specific, that the T-cell engagers have been very effective against hematological tumors, but not so much about with solid tumors. And as you say, we're starting to get there. But why do you think it's so hard? Is it the microenvironment? Is it accessibility? Is it targets?
Yeah, for me, based on what I've seen, and not only with Xencor's pipeline, but with external pipelines, there are a number of challenges to developing these. Cytokine release syndrome is one of them, but I think that is becoming a solved problem. It's still a highly empirical process. It's some art and some science to figuring this out, but as happens with most fields, it starts to become more science and less art over time as you see this working again and again and again. So I think that's a solved problem. And the basic approach there is, you have a low priming step that kind of tickles the T cells, conditions them a little bit, desensitizes them, and then you can start escalating on top of that. And you retain the anti-tumor effects of the T cells, but you actually blunt the cytokine response. And so that's, challenge number one, I think, is a solvable problem. I think the biggest challenge, I don't really think it's so much about the immune environment and whether or not there's enough T cells, because you see drugs like tarlatamab working in cancer types like small cell lung cancer that, at least by RNA expression levels, don't have a tremendous number of T cells compared to other histologies. The targets that seem to be working, particularly for T cell engagers, just happen to be highly selective targets, meaning they're selectively overexpressed in one or more tumor types, for the most part, one tumor type relative to all of the other normal tissues. And then coupled on top of that is having the right kind of selectivity engineering to take advantage of that selective overexpression so that you can more effectively engage the tumor cells versus the normal cells. And that's where platforms like ours and other approaches where you've got bivalent engagement of the tumor antigen and you can tune the affinity so that you maximize the selectivity of targeting the tumor cells versus the normal cells.
So I have a question about the whole TCE, T-cell engager concept, relies on pulling a T cell up to a tumor cell, activating it so the T cell kills that tumor cell. But that's what the T cell does. It kills that tumor cell. So if you have 1 million tumor cells and 1,000 of them, for whatever reason, don't have your target on it, and the rest of the million do, any histology you do, any tests you do will say this tumor is very positive for this target. You put it in your T-cell engagers and it could kill 999,000 cells, but the 1,000 that are left could presumably grow back without the target. So it's the escape mechanism, and so many cancer drugs have this problem where they work for a while and then the tumor comes back and it's resistant. How can we deal with that? Is there any way to get the immune system to not only kill the cell that it's attached to, but to kill another tumor cell, to get primed to kill other tumor cells even if they don't have that particular target?
Right. Yeah, Mike, it's a fascinating question. So one would hope that when you have a T-cell engager targeting a antigen positive cell, that you're also creating a lot of noise in there for the endogenous immune system to get some traction. So for example, all the T-cell engagers I am aware of, they all cause interferon gamma response. And interferon gamma does one thing very famously. It actually upregulates class 1 MHC on neighboring cells. And so the hope would be that you get kind of an immune bystander effect where you can heat up a tumor that started out being cold by targeting it with your T-cell engager. And in fact, we disclosed some promising, exciting data for our ENPP3 CD3 program. Now, it turns out that something like 90% of clear cell renal cell carcinoma patients express a high amount of ENPP3, but it's not 100%. So we didn't actually screen on ENPP3 expression, but we collected the data. And we talked about in our recent public disclosure, we had one patient whose ENPP3 positivity in his tumors was something like 20-something%. It might have been a little higher, I'm forgetting the number, but it was not 100% like most of the patients. Yet that patient was one of our responders. We saw a significant, certainly a PR, and again, I'm forgetting the exact numbers, but it was a very healthy response. So, the interferon gamma potentially heating things up, but also it can actually act at a distance because it's tumoricidal itself. We've seen this in vitro where we can incubate one of our T-cell engagers against target positive cells together with target negative cells, and we actually see killing of both cell populations.
Okay, thank you. That's a very good explanation. So thinking about bispecifics, but going beyond cancer, what modalities and disease areas do you think will dominate the market 10 years from now? Do you think it will still all be cancer or so much of it cancer, or will there be some other areas that become big?
Yeah, so it's been really interesting. So there's been a bit of a gold rush on two fronts. So one front is unlike 10, 15 years ago, there are multiple autoimmune diseases where there are more than one validated target. For instance, one of my colleagues, Kendra, talked the other day about making a TL1A IL-23 bispecific antibody. And sure enough, if you look back, this is for treatment of inflammatory bowel disease. If you look at various clinical trials, turns out IL-23s have a significant response rate in ulcerative colitis. The TL1As, none of which are approved yet, but they have extraordinarily promising clinical data, phase 2 data. They're now in phase 3. Anti-TL1A actually has significant impact. So that's an example where it's not rocket science. You've got two different targets. They're both clinically validated for that disease. And you hope that if you build them into one molecule, you can actually have greater impact of that disease. You can raise that ceiling in terms of response rates in humans. The other gold rush, had another question about sort of papers that have come out recently that are disruptive. I would say the most disruptive paper that came out in the last couple of years in our field was the Georg Schett paper talking about use of CD19 CAR T for treatment of autoimmune disease. right? Basically leading to a lot of hyperbolistic headlines, but basically the concept that treatment with the CD19 targeted CAR T could effectively put a number of lupus and Sjogren's and myositis patients into remission for long periods of time, kind of a immune reset hypothesis. Based on that paper, since we're talking about bispecific antibodies, there is another gold rush going on deploying T-cell engager bispecific antibodies where you're targeting a B-cell antigen. Like, in fact, Xencor's got a CD20, a CD19 program. Other folks are going after BCMA. The idea being that those B-cells are greatly contributing to the pathogenesis of these autoimmune diseases. And if we can target the right populations of B-cells, we can not only hopefully put that patient into remission because we're getting rid of that hyperactive component of the immune response, but if you translate from what Georg Schett had shown, potentially even resetting the immune system for a long period of time so that your pathogenic B-cell populations are now replaced by naive B-cells that aren't autoreactive.
Right. The question I have is whether you lose the immunities you've built up and you have to start over.
And in fact, it's a fun field to think about, learn about. So one example of that is thinking about the different targets and the risk benefit of the different targets. I know the people exploring BCMA CD3. BCMA is most highly expressed on plasma cells, and they are basically prospectively, when they dose their antibodies, they're actually giving the patients IVIG to effectively replace their antibody repertoire so that they have at least passive protection against pathogens. I think the real evolution that we're going to look for in that field is figuring out how much depth of B-cell depletion do you want. It's generally understood that the deeper the better in terms of patient responses, but maybe more importantly, after the depth is what's the durability of that depletion. And so can you find a sweet spot where you've got enough depth and durability of that B-cell depletion to do this immune reset, but also allow the immune system to recover and be able to react against, infectious pathogens.
Right. And the just to clarify, a plasma cell is a type of antibody producing cell that's job is just to produce lots and lots of antibodies. It's sort of the end of the line from early B cells, naive ones, and then B memory cells, and it goes down to plasma cells. So if you get rid of the plasma cells, you stop making antibodies, but you still would have the early B cells, right?
That's correct. But it all depends on who you target, right? So there's a number of people going after CD20, which is the more restrictive of the B cell lineages. CD19 is a little bit broader. BCMA goes far later, but can also capture a lot of the earlier stage B cells because it's actually expressed on those populations.
So we'll see how it works out. I'm actually really curious to see if bispecifics end up being useful in things like cardiovascular disease, or maybe we get better at getting them into the brain. Just because when you have, cancer is particularly distinguishable from other cells because it's rogue, whereas your heart cells aren't that different from other cells, but there are differences. And if you needed to go do something to a heart cell and you don't want to do it to any other muscle cells, if you could find that difference and make a bispecific or a trispecific that exploits that, you could maybe be very specific. I'll be very interested to see if that ends up happening.
Yeah, no, I'm with you. It's something, unfortunately, I don't know enough about those diseases to know how to tackle them yet, but I think you're right. I mean, the world's going to be looking forward to using these technologies and expanding their application. There's been a lot of application in, like you said, in oncology and autoimmune disease, but I think there's a lot more frontiers out there to tackle.
Right. And I'm with you. I don't know the heart biology at all. I just use that as an example. I think that part of it is just education. I think that people in those fields don't really think about antibodies as drugs and need to learn about how you can engineer antibodies to do these things. And then they might think of brilliant ways of using them. So, well, let's hope for that. Okay, enough of the technical. Now we can get to the lighter part of the podcast. So, John, tell us about your favorite project. What was it? What made it your favorite?
Yeah. I mean, I actually talked about it earlier. So obexelimab was probably my favorite project, at least back in those days when we were doing that work. I mean, it was just such a fascinating molecule in terms of, it was effectively a bispecific antibody because if your Fc is enhanced for engagement of an Fc gamma receptor, then you are engaging two things. And that was just so much fun to watch. And I'm still looking forward to seeing, how Zenus does with that molecule. These days, I would say my favorite project is our, we call it XmAb 819. It's our ENPP3 CD3 bispecific antibody that I mentioned earlier. That was a target that was discovered, we thought, was discovered internally. A guy named Alex Nistal had come in and was doing some bioinformatic work on the TCGA, looking for things that are selectively overexpressed. So he pulled out that target. Once we knew that it was an interesting target, we realized that a company named Agenus had made a drug conjugate against that. It didn't do well for reasons of payload toxicity, but they actually saw some tumor reductions, and it looked like they didn't have a lot of target off-tumor toxicity. They had sort of the generic toxicities like ocular toxicity that comes with certain payloads. So we tackled that, and we were able to use what we call our XmAb 2+1 platform, which has bivalent engagement to give us more selectivity, and then finally gotten in the clinic. And, now we're up to, as can happen with these T-cell engagers, because as I mentioned earlier, you have the priming step-up strategies, and there's a lot of art and science to figuring those out. We're up to cohort 10 because you also have to start with these things at what's called a MABEL dose, the minimal anticipated biological effect level, where it's effectively a homeopathic dose of your drug. And so
it just leaves like the impression of the drug in the water.
Exactly,
right?
Exactly. So it could take some time to dose escalate. But now, with our latest data release, we're really having impact on patients. The kidney cancer doctors are super enthusiastic about this. One of our lead clinicians is Monty Paul at City of Hope, and he's super fired up because these patients, once they get to this stage, they've already progressed after getting either Ipi-Nivo or, which is a dual checkpoint inhibitor, or checkpoint plus tyrosine kinase inhibitor. Now they're also getting this HIF-2 alpha inhibitor called belzutifan. And so, we're seeing responses in patients that have had 3 to 7 prior lines of therapy. And there's really nothing else out there of this class. So I would say nowadays that is my new favorite project.
Well, that's very encouraging that it's looking so good, and best of luck with that. On that note, we can get ready to wrap up. And as we come to the end of each podcast, I like to ask our guests to share something they've recently discovered or enjoyed. It might be a book, a film, a piece of music, memorable place, another podcast, anything you think our audience would find intriguing or inspiring.
Yeah, sure. So as some of you know, I've always had to have a creative outlet, and that's often been music. But more recently, that has evolved into becoming passionate about landscape photography. In fact, I bought myself a Jeep Grand Cherokee so I could off-road to my heart's content to get to the good photo spots. And so, in fact, just last weekend, I was at a place called Alabama Hills. It's one of my favorite places in the world. I was there photography-wise, particularly because I wanted to be there for what's called the alpenglow. That's when you get that morning pre-sunrise light that just turns the Sierras kind of this pinkish color. So I was at that spot. But I did notice that I'd gotten there late on a Friday night. So my 5 a.m. wake up was going to be Saturday morning the next morning. But when I was pulling into Lone Pine, where this is all located, this is on the 395 east of the Sierras, there was almost a full moon out. And when you've got snow on those peaks in the Sierras, they're just glowing at night. And so I told my Labradoodle, who's my constant photo companion, I said, Cody, tomorrow night we're going to do some moonlight shots. And I would say that the most special moment for me, aside from being up in a beautiful place like Alabama Hills, where you're all alone, there's nobody for miles and miles, and you're way up on top of this rock outcropping. You're sitting there, trying to get your camera set up in the tripod. But the most special moment was just seeing my dog, Cody, sitting there, just looking like he was completely in his happy place. Cool mountain air, moonlight, could see for miles, and just looking around, being completely content up there with me. And, we really had a nice trip. And in fact, I had to give a board member a ride the other day in my car. So I had to rinse all the mud off of it from our off-roading excursions on that weekend trip.
Well, thank you, John. What a wonderful, great story to end this episode. Thanks for sharing your stories and science with all of us. And thank you all for the important contributions you and your team at Xencor have made for the therapeutic antibody field. So that's all for today's episode of Antibodies. Thank you so much for joining us. If you'd like to learn more about the Antibody Society, just head to antibodysociety.org. Keep an eye out for future episodes. We've got more fantastic guests lined up. We always appreciate your thoughts and suggestions. So if you have feedback or ideas for topics or guests, feel free to reach out using the link on the Society's homepage. We'd love to hear from you. And so John and I might do a little musical outro with our harmonicas. And if you enjoyed our chat, make sure to subscribe so you don't miss what's coming up next. And if you feel inspired, share us with a colleague or leave a quick review. It means a lot. Until next time, I'm Jonathan Davis with Creative Antibodies. Keep exploring, keep discovering, and stay connected.
Yeah, thanks, Jonathan. It was a lot of fun being here and doing this with you.
My pleasure. That's all for today's episode of Antibodies. Thank you very much for listening. I hope you can join us for our next episode. Meanwhile, check out all the materials and announcements relating to antibodies, including information about our sister podcasts, at the website antibodysociety.org. The executive producers of Antibodies are Kristen McPike and Josh Dorai, with additional production and ideation help from the many volunteer members of the Meetings Committee of the Antibody Society. Editing and original theme music is by Jeremiah Davis. Antibodies is a production of the Antibody Society.
That's good. Awesome.